So with that, I'd like to move us to the next section of our agenda, where we're moving into to look at some of the activities and different perspectives around manufacturing, the implementation of innovative manufacturing technologies across the medicines ecosystem. Again, we've got 3 excellent speakers. We'll have a Q&A session at the end, so please do pop your questions into the Q&A on Slido as we go through. In the interest of trying to manage the time and give you as much opportunity to hear from the speakers as possible, I'm going to give the 3 speakers a brief introduction now, and then we'll go through their presentations in turn ahead of the Q&A. So the first of our 3 speakers is Professor Gavin Halbert. He's actually one of my colleagues from the University of Strathclyde, where he's also, as well as being involved in CMAC, is Director of the Cancer Research UK Formulation Unit there. It's a wide range of interests spanning medicinal analytical chemistry, formulation, manufacturing technology for both parenteral oral products, and actually runs a GMP formulation unit within the university campus. And Gavin's going to look at medicines innovation from the academic research perspective. After Gavin, we have Dave Tudor. Dave, as many of you will know, is Managing Director of the Medicines Manufacturing Innovation Centre, and will highlight the pre-competitive approaches, again building on our themes around collaboration and working across academia, industry and government, to highlight the technology opportunities and the grand challenge business models that are being implemented within MMIC to help, as Clive highlighted, with the standardisation agenda, how to democratise the technology and get that into the hands of commercial organisations. I think also touching on regulators and the need for alignment there to give the benefits to patients and the economy. And then our final speaker in this session is Ian Shott. Ian is Managing Partner of Shott Arcanova and leads a portfolio of life sciences businesses, looking at how to accelerate growth and improve performance across those manufacturing activities. He's also the co-founder and executive chairman of Arcanova, very involved in contract research and development business based in the UK. So brings, I think, a different perspective, commercial perspective, in terms of the opportunities and challenges for advanced manufacturing. And he's going to talk about the use of continuous process technology and biocatalysis to streamline the latest small molecule API therapies. So that's an introduction to our speakers, and with that, I would like to hand over to the first of those, Gavin Halbert.
Thank you very much, Alistair, and welcome everybody. It's nice to be here today. Thanks to the organisers for inviting me. What I'm going to try and do is to give you an insight into some of the academic issues associated with medicines innovation, and the sector that I work in is quite definitely academic and or small molecule and or spin out type companies. So we're not sitting here with a large pharma behind us. And I'm also, as Alistair said, sort of a member of CMAC. So I'm going to include some of that in what I'm going to talk about as well. And this particular slide was almost teed up by Jonathan. So thank you, Jonathan. And we haven't really discussed this, so it's thinking along the same lines, I think. So for me, medicines innovation starts with that first administration of a medicinal product to, in the cancer chemotherapy zone, it's usually a patient, but obviously could be a volunteer. But that medicines, that single tablet, single injection, doesn't simply arise out of nowhere. There is a whole pile of research that zones in to give us that particular medicine. That might be small molecule. It could be some of the issues that Jonathan has mentioned about biotech or advanced therapy medicinal products. So that research has to be there. But that research has to be translated into a product. So you have to have a product that you can actually administer to the patient. And that in itself brings with it a range of restrictions. So you've got the restrictions of the research, you have restrictions of the translation to get to the product. And then, as Jonathan's already teed me up, you've got the restrictions of the patient. Now, within the cancer chemotherapy field, personalized medicine or directed therapies are what we aim for. And one of the most recent projects that we looked at had a prediction for 300 patients a year in the UK. Standard processes really are not going to be able to deal with that type of level of demand. And finally, you've got to implement that particular product, project, or medical innovation. Now, that implementation can come unstuck for a whole range of reasons. Scale up is a very common issue in academic projects where the inventors haven't really thought about how you might get from the bench to the bedside. And equally, you've also got to take account of regulatory issues. And the particular graph there is one that I do like showing because it's the weight of the orange guide with time. And I really, for an industry that's supposedly at the leading forefront of everything, to be sitting on what is an exponential curve, I find is very bizarre. And given the comments about net zero, it's almost, I'm tempted to give that curve to Greta, and I think she would be after us all for our lack of control. I had a very early lesson in this back in the 1990s when I took over the unit with a drug called temozolomide. We put that into phase 1-2 clinical trial, and it became very obvious very quickly that it was working. It was for the treatment of malignant glioma, which is a condition that is realistically a condition of an unmet need. This was the only drug that was actually giving patients a chance of sustained life. And what happened was I ended up with the editor of the Daily Telegraph and the Daily Mail chasing me because they found out I was the only person in the UK with supplies of the drug. In fact, I was the only person on the planet with supplies of the drug. But we'd blown it. We'd blown it because the synthesis had a diazo intermediate. For those of you who can remember your chemistry, they have a tendency to go bang. The chemists would only ever make 100 grams of this intermediate at a time, which then meant that we couldn't actually put the drug through a standard process. And within the unit, we ended up hand filling capsules simply in order to meet the demand and to meet the scale. And this to me was a very early example, although I probably didn't know it at the time, of us developing a continuous process. So the chemists were synthesizing. The next week we had the drug, put it into capsules, and the week after it went out to the patients at the various clinical centres in the UK. But it was a very early learning lesson for me in that I really needed to look at what we were doing and always, always, always have the ability to push the accelerator. So if the drug works in cancer, people are going to want it, and they're going to want it very quickly. So one of the things, obviously, a gratuitous quote from Einstein is that we can't expect to do the same things and actually resolve those problems. So what I'm going to cover in the next part are 4 areas where we're using continuous, some of which are applicable now, and the end one, which actually might be something that would come in the future. But equally, that gratuitous photograph of Albert Einstein is really just to point out that he was obviously well ahead of the curve, as everybody knows. But this really is probably an example of a lockdown haircut rather than a theory of relativity. So, nanomedicines. Nanomedicines are a personal interest. It was an area I did my PhD in. And it's an area where we've had multiple presentations in the cancer chemotherapy field, but we've always failed because they've simply not been scalable. What you can do on the bench to treat a mouse is very simple, but to actually try and scale that, along with all of the other requirements that you have, was actually very difficult. We have recently been looking at microfluidic technology for the production of these nanomedicines. So the picture there on the top right is a a microfluidic chip, and it's a continuous process. You put two fluids in, the chip mixes the two fluids, and the output is your product. So it's continuous and it's scalable, and you can get from research scale to GMP scale. And the graph in the middle is part of recent research that we've been doing, where we were examining this particular process and we were preparing liposomes. And you can see that the scale was going from the bench scale, which is the 2 histograms in the middle, through to 60 mils per minute, through to 200 mils per minute, which was the GMP scale process. And you can see that you can scale this process very simply and very easily. We have applied that to liposomes. So we made liposomal doxorubicin. I saw a question from Liz Meehan. We could also apply it to multiple other parenteral systems. And we've also been using it to produce RNA vaccines using lipid nanoparticles. So an incredibly versatile technology. It's a bit clunky because you still end up having to use standard post-formation processing techniques, and that's not brilliant. And we've also been looking at applying it to biotechnology applications, the sort of applications that Jonathan was talking about earlier. So it's there, it's usable, and it actually could be implemented now. And it's a continuous technology. Oops, my internet clicker has gone. The other thing that we've been looking at, 3D printed oral dosage forms. Now, we are not unique in this. There are multiple groups looking at this around about the world. Clive mentioned it in his introduction. And again, it uses continuous technology. The device that is heading to you out of the screen is a hot melt extruder or twin screw extruder. That's a continuous process. You pour the powder in one end, and in one version of it, out the other end comes a strand of polymer and drug. And that polymer and drug can then be used in a sort of standard, what's called fused deposition modeling 3D printer to give you a range of tablets. Now, this process is actually very cheap. The 3D printers are almost disposable. And there's an increasing acceptance in terms of their use. There are now multiple licensed products available on the market. But whilst that is incredibly useful, the inherent within it is an amazing amount of ability to vary. And I'm not going to try and sort of get into a lot of this, but in the screen at the bottom, there's a range of strands of drug and polymer. It's paracetamol in afinisol, and it goes from low concentrations, it looks a bit blurred on my screen here, up to high concentrations, and around about 40%, you can see that the strand starts to go white. Now, the reason it starts to go white is because the paracetamol, or acetaminophen for our more international listeners, is present in the solid form, whereas in the clearer strands, the paracetamol is actually dissolved in the polymer as an amorphous phase. So if you're going to try and make a medicine out of this particular system, you've actually got to get control of that process of the drug in the polymer. So whilst it's incredibly adaptable and flexible, that brings additional challenges in terms of the actual formulation of the process. And the other big challenge, which Dave Tudor is going to cover, is that the pharmaceutical industry has been making tablets for quite a long time. So unlike the previous example with liposomes, this example, whilst it's new technology, it's continuous, has got an obvious competitor within the industry in terms of the standard compressed powder tablet, which probably means that whilst the liposome example could be instantly adopted and used, this example is probably going to have to find a niche where it can actually be applied and used sensibly within the industry. Where is that niche? Well, I'm very interested in this because in my end of the business, clinical trials, you do start with small numbers of tablets. It's that one tablet that you put into the patient. So there's an obvious niche application of 3D printing in clinical trial medications. Right, the next type of approach you could look at, which we are looking at within CMAC, and I know colleagues within MIT are looking at and have published on this, is the microfactory. The two previous examples required preformed API or drug, and it was realistically a secondary process that was producing the product. In the microfactory, we are trying to look at the end-to-end approach so that in the system, the microfactory, you produce both the drug substance and the drug product. And we're trying to look at this as being on a benchtop scale. So in other words, something that you can see the sort of photograph there of one of the CMAC labs, something that fits in a very small footprint, something that Jonathan mentioned is good for reducing sort of carbon costs or emissions. But also, if you think on it, if you think back to my temozolamide's example, if you can only have a small amount of material in any one place at any one time, this would have been the perfect solution for my problem 30 years ago. And it's probably also, therefore, the perfect solution for some of, again, these small use clinical trial type areas that I work in. It's probably not quite yet the be all and the end all, because I don't think we've quite sorted out the synthetic chemistry. But it is something that is probably going to expand and will be of benefit. We have looked at the other end of the process, so the printing of the product, and there's just an example result there of the blue, which is the dissolution from a tablet that we've printed as output from a microfactory, compared to the current commercial formulation, which is a standard powdered capsule formulation. And you can see, and it echoes what Jonathan was saying, that there's an improved quality and consistency within our 3D printed microfactory product. The last thing is quite definitely something that's not yet there. So it's realistically research that we are currently looking at, and it's non-destructive release testing. So in this particular example, we have taken an ibuprofen tablet, 20 milligram caplet, and we've potentially thrown the analytical kitchen sink at it. It probably would have been easier to throw it at the kitchen sink. But a brief in terms of what we've done, we've taken the tablet, we've performed an X-ray nano-CT of it, so that gives us an internal view of the tablet. We've then deconstructed the tablet by taking away layers, analyzing each layer for individual content. And then once we've got that individual content layer by layer, we can then reconstruct the tablet to give us a three-dimensional representation or map of everything that is in the tablet. It also allows us to calibrate the X-ray or nano-CT so that we can do that without having to destroy the tablet. And once you have that calibration, you can then analyze individual tablets. And for those of you that know anything about tablets, the easiest way to look at that is the sort of panel at the top, which has got titanium dioxide. Titanium dioxide is a component of the tablet coat. And if you look at the bottom part, you can see that it forms a ring around the caplet. The other ingredients, as you can see, are in very much a random mixture. But if I know enough about the tablet, and this potentially is where the modeling and digital control comes in, with that X-ray analysis of the tablet, it actually provide me with enough information to say I've analyzed the tablet. Would it tell me all the critical quality attributes? It would definitely tell you the amount of ibuprofen that's present or the drug, so you've got the content of the active. If you know enough about how the tablet is arranged and you can model that, will it also tell me about disintegration and dissolution? That maybe is a drawback of the method of manufacturing a tablet, because you're always going to get this random mixture. So maybe that is where the three-dimensional printing would come in, because you would then have a tablet with a slightly more consistent structure than a powdered tablet produced by normal mechanisms. So, sum up, where do we need to be? Well, I mean, I think you're probably going to hear a lot of the same from a lot of the speakers this afternoon. We potentially need to actually be able to think from end to end. And by end to end, I mean the molecule and the disease right the way through to the patient. And the sort of staff of caduceus that's tipped on its side is the sort of small representation that we put into the CMAC hub grant. And I think it represents realistically what we've got to do. We've got to get a hold of the process. We've got to get a hold of the product or the drug, and we have to integrate that to give us a pathway that leads to the patient. I've probably not thought this slide out enough, but it's realistically to go for our last speaker in this session, and that's one of his boardrooms on the left-hand side. He can explain where that came from. But the book in the middle is a prescription book from a pharmacy that was in Glasgow. And the prescription example is from 1897. And realistically, what we're probably looking at is what they would call mass customization. I don't really like that term. I'm not sure the term digital pharmaceutical artisan is better, but realistically, we are going to return to that area for some therapies, not all therapies, where there's an element of skill and combination of resources to allow you to produce a product that is individual to the patient. So with that, I will finish. Thank you for your attention, and please stay safe.
Thanks very much, Gavin, for that excellent overview and I guess a snapshot of the multiplicity of approaches that are being explored, in your own research, and of course, echoes across I think many of the research groups that will be listening. With that, we'll hand over to our next speaker, so Dave Tudor, who's going to present on accelerating advanced technology translation. Dave.
Alistair, thank you very much for the kind introduction, and I'm really delighted to be here this afternoon to talk at the symposium. I know, there we go, we've got the first slide up, good. I've spent almost a full 29 years in the pharmaceutical industry, and I wanted to use this topic today to talk to you about some of my experiences, some of my thoughts about how you can accelerate advanced and difficult technology into the industry. I spent many years as the head of API within GSK and some years as the head of strategy, and so I've been involved in many discussions and many business reviews about how you bring new technology into this industry. And if I flick on to my first slide here, I think it's fair to say that the pharmaceutical sector, I think at long last, over the last 3 to 5 years, is waking up to the value of technology. And in particular, we're gathering momentum. There is no doubt about that. We can see evidence of more and more advanced therapies with advanced technologies coming into the marketplace, adding value to the patient, adding value in terms of cheaper pricing, and adding value in terms of a better carbon footprint. And what I wanted to do today was talk to you about really what I think the magic ingredient, let's call it the magic sauce, of how you can bring technology in a faster way. And that is in and around, are you prepared to go into a partnership? Are you prepared to go into a technology collaboration? And do you want to share the risk? The picture I had in my mind when I was preparing these slides, it's a bit like comparing just now pharmaceuticals to the farming sector. Are you going to be that farmer that ploughs his field in his own ways done for the last 100 years with his own old tractor and his ways of doing things and his experience or her experience? Or are you going to be that collaboration of farmers that are helping each other out, trying to get to the same end goal, using advanced technologies and sharing the cost and the risk of that technology? That's an interesting question. So as we go through these slides, ask yourself the question, which farmer are you going to be in terms of accelerating technology in this industry? Now, to do that, I'd like to talk to you about the Medicines Manufacturing Innovation Partnership, which is really a combination of two wonderful things. It's a technology business model, which we call the Grand Challenge model, and it is really focused in on trying to identify in partnership with the industry future disruptive technologies. Those technologies can be around modularity, for example, continuous manufacturing that allows that to happen, certainly looking at digital in terms of both design and manufacture and supply, and advanced technologies in and around analytics to allow better control and better understanding. And last but not least, how can we connect the supply chain? How do we get these independent vertical vats of data and get them talking to each other to add value? And so that's what we mean by disruptive. That's what the MMIC business model is about. Can we identify these big challenges? And then can we form collaborations? Can we form the right group of companies that are going to get a benefit from this technology, the right group of partners that can help to unlock the technology and get the right ecosystem together to do it with pace, momentum, and focus? That's really what MMIC is all about. Now, rather than me take you through some slides about the rest of MMIC, I'm now just going to let this, hopefully, technology will play a little video.
Aging populations with increasingly complicated health problems, combined with rising costs for drugs and a need for more personalised treatment, are just some of the challenges facing the pharmaceutical industry today. The Medicines Manufacturing Innovation Centre aims to address these challenges by creating more productive ways of manufacturing medicines and developing an agile pharmaceutical supply chain for the future, which will reduce waste, costs, and speed up time to market, enabling a healthier society and a robust UK economy. The centre is a collaboration between CPI, the University of Strathclyde, UK Research and Innovation, Scottish Enterprise, and founding industry partners AstraZeneca and GSK. The facility, based in Glasgow, Scotland, is currently under construction and is due for completion in late 2021. It's a unique, state-of-the-art GMP facility which will enable industry, academia, healthcare providers and regulators to work collaboratively to de-risk new technologies, providing a clear pathway for their widespread adoption within the pharmaceutical industry. The initial focus of the centre is to deliver two grand challenges. Grand Challenge 1, in partnership with CMAC, focuses on redefining the production of oral solid dose medicines. Advanced technology will enable tablets to be produced 30% more efficiently versus the current industry standard. Grand Challenge 2, in partnership with CME, uses digital quality and automated technology to transform just-in-time medicines supply. This next-generation technology will reduce waste whilst improving flexibility through clinical trials, leading to a dramatically quicker supply of drugs to patients. This is just the start. Two new grand challenges will be launched in the coming months, designed to be both ambitious and disruptive. They will support the pharmaceutical industry to develop step-change technology to make the manufacturing and supply of drugs vastly more productive and with reduced risk. Further grand challenges will follow on from these. We are delighted to welcome several new collaborators, and we continue to expand, seeking new partners to join us in developing next-generation pharmaceutical manufacturing. So why not be part of this innovation journey? Let's innovate together.
So hopefully that video explained what MMIC is about. I want to just finish up the introduction to MMIC by just finishing talking about the operating model. The model is that we create partnerships, this triple helix of industry, government and academia working together, focused in on ambitious grand challenges. You've seen in the video GC1 and GC2, and I'm going to talk a lot more to bring the partnership to life on Grand Challenge 1, which is on continuous direct compression, but you may have heard 2 weeks ago about the new launch of the oligonucleotide efficiency Grand Challenge. And then what's really important here is that the industry want us to commercialise this technology. So we're not just doing a development step. We're going to take the technology, working with the regulator, demonstrate it, gain the GMP license for the technology, build the industry capability and skills such that the industry can literally just lift and cut this in where appropriate. Let me just get the next slide on if you don't mind. Thank you. Yeah. And my final slide here, just by way of introduction, is to really just position where MD sits in the TRL ladder. We are very much focused in on the commercialisation, the scale up, scale out, and commercialisation of the technology. And we can do that in a number of different ways through partnership-driven research, fee-for-service approaches, or public funding research programmes. But just as important as that is that we need to make sure we find the right partners to deliver and to execute the technology. And that's the early TRL 1 to 5. And we're very pleased with the partners that we're already developing. And in particular, I want to call out today the CMAC organisation, who are doing a super job on Grand Challenge 1. Now, if I can move on to the next slide, I want to really bring this partnership approach to life with, as I say, this accelerating continuous threat compression. Next slide, please. So Grand Challenge 1, what is Grand Challenge 1 trying to do? Well, we know that continuous direct compression as a technology has been in and around our industry now for about 10 years. We also know that there's only a handful of drugs in the market through CDC. So why is that? Why is this technology that offers us big improvements in cost and efficiency? It allows us to address this challenge of the pipeline with smaller volume, higher potent products. We can make smaller batch sizes with better control. And it allows us to really drive digital control and modular factories and avoid the cost of capital, which is a big issue for the industry just now. So why is it this technology isn't being driven or isn't evolving at the pace we would like it to go? And if you actually think about those benefits I've described, if you just look at the UK economy, then we know that the most productive part of the UK economy is the export of pharmaceutical products. About 70% of that is tablets that are made in the UK, shipped elsewhere around the world. And if we don't improve the technology how we do tablets, then we're going to run into long-term life cycle economic problems with that sector, with that industry. So it's important we transform the technology and we continue to drive value in and around how we make tablets. If you go on to the next slide. So these are the objectives of Grand Challenge 1. We've got 3 very, very clear objectives that we want to achieve. The first one is that we want to try and pull data sets from different partners, whether you're a technology company, an industry partner, or an academic partner. And we want to pull that data so that we can actually develop predictive digital data twins. One of the reasons why this technology doesn't get pushed as much as it does is because at the early development stage, companies have got the choice between batch or continuous. And that's a big decision to make. Many companies end up trying to do parallel development programs. So we can take data sets and API particle size information and we can start to predict the confidence of CDC. We can help those development decisions and help them to translate and bring this technology into the marketplace. The second objective is really trying to look at understanding the science, solid state chemistry, and how we do CDC. There's still a lot to learn. There's a lot to monitor. So can we bring some advanced analytical techniques in and really build a control strategy that helps us to understand step by step what's going on, build confidence with the regulator, and make sure we use that data to affect a real-time QP release strategy. And the third objective is to try and modularize the equipment. Now, what's happening today, many of the engineering companies that ship into the industry now sell machines that are connected together with their own control systems, and they're very complex pieces of kit. The industry needs flexibility. It needs to modularize the different suppliers in terms of equipment. It needs to allow to evolve and drive the control systems itself. So we're trying to get standardized hardware design and control strategy for CDC. 3 very big and very ambitious goals for this particular technology. And if you go on to the next slide, I'm not going to touch on all these bullet points because I'm sure you can read them as quick as I can. I want to touch on a couple of them if that's okay. This idea of being able to really use the data to predict the future, the historical data that we generate in the industry, by taking the risk and sharing that data together in a pre-competitive way, can we help to predict the future? Well, the answer is yes, of course we can. So what is stopping us doing it? And in this project, with the partners that have signed up, and I'll share with you them in a second, that's exactly what we're doing. And we would welcome other partners to come and join. The second thing is in around this decision about, is it a fully connected CDC system, or is it a combination of mini batch and continuous manufacturing? Well, how can we model that? How can we help to predict what the right thing to do is there? And so the hardware that we're designing has both got CDC and mini batch in not just the hardware and analytical control, but also in the digital twin that we're generating. And the last thing I wanted to highlight here is to make sure that we develop the appropriate control strategies. There's lots of great ideas, and there are some companies that are doing some amazing things, but the control strategies here are really important. If we can define and build our confidence, work with the regulator in the control strategies, then there is no reason why this technology can't be the premier choice of tablet manufacturer for the industry going forward, whether that be a brand new process coming out of the R&D organisations, or whether that's a retrofit into existing products that are on the market today. Now, my next slide, and but last, is really just to show the extent of the collaboration. So to make this happen, if you can move the slides down one, thanks. You can see here that the partnership model is growing, it's evolving. So there's no one organisation on this piece of paper that can deliver these ambitious goals on their own. But by bringing together pharmaceutical companies, and GSK and AstraZeneca are at the heart of this, but there are other pharma companies that we're about to announce joining. By bringing the right academic and technology organisations together, by working with the government agencies and working with technology companies, bringing ideas from other sectors like applied materials from the semiconductor industry, by bringing all of this together, we can unlock significant technology challenges. We can do it cheaper than individual companies doing it because you share the cost risk. We believe we can do it faster, and we believe we can share the pool of resources and experience to drive the optimal outcome. There is no limit to the ambition about how this model works. So my last slide before I hand over to Alistair, if that's okay, I'll go back to my original question. What kind of farmer are you going to be? The tried and trusted way of working on your own, driving the technology, using your expertise works. And that drives at a certain pace, at a certain cost, with a certain risk. But when these farmers pull their technology together, they share and help each other, they plough that field.
Well, it looks like we've maybe just had a glitch there with Dave's connection. However, I think, it's ending on a call to arms in terms of the collaborative agenda, and again a recurrent theme. So we've heard around the driver and activities in the research space and of course in the innovation space. And I guess our final speaker in this session, Ian Shott, I guess brings us to the sharp end in terms of industrialising advanced technology. So I'd like to hand over to Ian.
Thank you, Alistair, and hello to everyone on this conference. I've got a deck of charts that I'm going to scamper through, but before going into that, I just want to set the scene. So I'm going to talk about the journey of Arcanova, and we created Arcanova by buying an unwanted site from Covance exactly 5 years ago. That site was operating in a preclinical arena and had a vivarium. We didn't take the vivarium, and we came up with a completely different strategy to focus on clinical development of new pharmaceuticals and small molecule pharmaceuticals. And on one site to have the capabilities of drug substance synthesis from discovery through the clinic to commercial in the case of very small volume pharmaceuticals. The second capability is to move through pre-formulation to formulation and into capsules or vials of drug product to get them into patient. And the third capability is to analyze those patients using bioanalytical techniques. So under one roof, to put the full supply chain from drug discovery to final product to improving the health of patients. Now, it's been said that I'm going to put the sharp end on this, and I've been involved in active pharmaceutical ingredient manufacture for 4 decades. And for 3 of those decades, I've run global businesses, either for multinationals such as AstraZeneca, ICI, Rhodia, or Lonza, or in the last 20 years for a portfolio of SMEs, of which Arcanova became the biggest. The CV or bio that was read out by Alistair is a little out of date, as last week we sold Arcanova to Quotient Biosciences, backed by the huge private equity company Permira. And my new role through Shott Trinova is to be the senior advisor to the board of Quotient Sciences, taking the strategy forward. Moving now to Arcanova, and although this is with a Shott Trinova heading, the rest of the talk is going to focus on what we have actually done in Arcanova. And I speak as a chemical engineer with manufacturing experience, and my priorities of focus have been on manufacturability, on materials efficiency, and on speed and time. I mentioned that we're focusing on small molecules, and despite the very exciting technology advances in gene and cell therapy or in protein chemistry, small molecules are still dominating the list of new drugs every year, and over 60% of those new drugs are small molecules. As other speakers have highlighted, the tendency for greater focus, precision pharmaceuticals, unmet needs in cancer are leading to a large prevalence of small volume, highly specific actives for these new pharmaceutical therapies. And our focus in Arcanova was to be capable of doing the whole journey to commercial manufacture at small volume. Now, when I talk about materials efficiency, I can look around the whole planet, and I don't believe there is one manufacturing chain for a pharmaceutical where more than 1% of the materials employed manifest themselves in the final product. So it's less than 1%. The other concept on time is velocity ratio, and that is the time of real value add over the total asset time available in a 365-day year on a 24-hour day. And I've never seen a velocity ratio above 10%, and usually considerably smaller. So the scope for improvement here is huge, and it's nothing like just trying to improve stage yield. We're talking about changing the whole process route as well as accelerating. I'm hoping now to advance the chart. Right. So we looked at creating the journey to full manufacture as a project. And we've been lucky enough to win support from Innovate UK for that project, working in close collaboration with Nottingham University on Continuous in a project that we entitled Flow Innova. But the focus of the project is to really make flexible continuous manufacturing a reality, and to be able to produce multi-kilo quantities or multi-tens of kilo quantities in essentially a development environment. And by doing that, we significantly reduce the time and the cost of both the development and the scale up and manufacture. As a second thread, we're looking at the use of engineering biology, synthetic biology, industrial biotechnology in certain areas to drastically reduce the number of transformation stages. which really does start to make a massive impact on minimizing solvent use, energy use, reducing the purification load, and above all, waste. Now, in our bones is a strong urge to collaborate, and we think that we can get places faster with very clear collaboration strategies. I was involved in creating CPI almost 20 years ago. I was also on the governing board of Innovate UK. So I've been pretty involved in trying to make this type of network function. And I've got iBioIC CPI, which are vehicles I've been actively involved in, and through that been close to Strathclyde University and CMAC, and recently MMIC. And we've been working with Dave Tudor to see if we can in some ways operate in a collaborative manner with MMIC. So core to our progress has been a collaboration model, also with specialist companies like Biocatalysts in Wales or Ingenza in Scotland. And what we've been trying to do is to look at how we can drastically change different processes. Now, this chart comes from the project I've mentioned. I'm not going to read it. We're short of time, so I'm sure most people can read it quicker than I can. But the point I'm trying to make here is that there are many different stages. We're trying to achieve very specific, real milestones with real products. And the other thing we've done is put the lens of Arcanova on this. So all our applications have been real customer projects that have been brought to us from our customer base of 250 companies spread around the world. Our focus is very simple. We are, above all, trying to simplify, intensify, and accelerate in order to generate better productivity, lower costs, and above all, better and improved patient care, and particularly getting to patients suffering in areas of unmet need. What we do on a daily basis, and various speakers have talked about data-driven processes, is to look at candidates coming out of discovery, and where possible, even trying to help with the selection of the selected candidate. We think that historically, this is often based purely on the basis of best drugability. And we think it's important to also consider some of the physical delivery issues and the manufacturing issues and the manufacturability issues. So we're trying to have an influence upstream, as well as make sure we're using the best data-driven processes to look at route development or new route scouting in order to get to a flexible manufacturing strategy. So here's a real example. It's somewhat generic. I put up, this is the only chart with what I would call chicken wire. And the reason I've put it up is to demonstrate the types of chemistry that we're talking about. Some of those are quite nasty. Some of those involve higher temperatures, and we end up with many stages. From here on, those stages will only have numbers and not names. And I'm going to focus in on some real work that was done. It was an 8-stage process of chemistry. The challenge was to make a kilo using 20-liter streams. We re-equipped a standard laboratory with floor-to-ceiling reach-in fume cupboards where we could assemble either continuous or batch synthesis trains of reactors. And we've used those reactors where, if it's in batch mode, 20 liters was the largest vessel size. And this process started off as 5 batches per stage, total of 40 batches to get the kilo, and a 40-week program. And most pharmaceutical manufacture involves multi-months, if not more than a year, in terms of total manufacturing lead time, rather than weeks or days. There are issues of scalability, issues of efficiency, issues of waste. And what we did was quickly develop the process to continuous and employed some bespoke continuous reactors, which we purchased. And that had the impact of making a very substantial change to yield, reducing the batch count by a factor of 3, reducing the time by a factor of almost 4, and reducing the materials usage and therefore, in consequence, the waste generation by a factor of almost 2. So very substantial benefits. As Alistair even mentioned, we are interested in biotechnology. One can use self-designed and self-manufactured biocatalysts in a conventional chemistry environment, or we could be thinking of more sophisticated synthetic biology and trying to be much more ambitious in eliminating stages and reducing the pathway. It's quite unlikely to go in one step from a easily available raw material to the final product. It's possible, but there may be a chemical step along the way. We don't believe in panaceas of either continuous manufacture or biotechnology, and think that on the basis of thermodynamics and kinetics, one needs to pick the best mix of capabilities in order to industrialize. We spent quite a lot of time looking at different modular systems, which I'm going to skip through. And what we've tried to do is come up with a strategy based on a finite number of process steps and the ability to reconfigure those steps quickly in appropriate environments to make a full range of different products. And now I come to, sorry, the last chart just emphasizes the multidisciplinary nature of activity, and we're lucky in the Arcanova business to have all those capabilities on one site and the ability to think back from the patient to the beginning of process conception and hence avoid quite a lot of the problems that generally happen in terms of difficulty in manufacture or difficulty in formulation. Now this chart looks at our actual building, and it looks like a tiny little toy. The diagram at the bottom is about 200 meters long left to right and 100 meters deep from top to bottom. And the area that's exposed was a vivarium, but we are changing that into a manufacturing scale-up area and trying to take advantage of the infrastructure and the overhead air handling to minimize the capital cost and minimize the time of equipping for continuous manufacture and scale up, getting us as far as commercial manufacture. The top, this is a two-dimensional diagram of that part of the building, and the top corridor in green we are turning into a drug substance arena. The blue area would be drug product. The pink areas down the left are the necessary laboratories, either for process control or quality control. And the project for drug substances was approved last year. It's under construction and it will be in operation in Q4 this year. Total project length, about 12 months. That's just a quick layout. I need to come to an end. I've exceeded my time, but the point I'm trying to make here is that so far we have not walked away from any opportunity to develop continuous process, to scale up, and we're even looking now at getting the commercial license and making commercial quantities within the next 2 years. I'll end there and join the joint Q&A.
Thanks very much, Ian. I think you've painted a vivid picture, I think, of what rapid implementation of innovation, can look like. So that's really fantastic to see. So we have a few minutes for Q&A, some questions coming in from the audience, and would encourage everyone to also respond to the polls, as well as please do submit your questions. Maybe just to start, if I can direct a question to yourself, Gavin, you highlighted one of the nanomedicine examples with the liposomes that you're ready to go. I just wonder, so what's the barrier? If it's ready to go, why aren't you going?
Good question, Alistair. So good question, Alistair. I mean, it's a sort of technology that we have put in our clean room. So we have been making 2 GMP RNA lipid nanoparticle vaccines. And if we had a project that was within our remit that required that technology, we would go. But as I say, we've developed the approach to produce essentially a generic liposomal doxorubicin. But I think I would come back to what Dave was saying, in that that's not really our role to manufacture and commercialize a liposomal doxorubicin. So I think it is the technology's there. We've used it. We've got a handle on it. We've got that skill set. But in terms of us just running out there and just commercializing any Tom, Dick, and liposome that we can find, that's not really our role.
So thanks. So Dave, I guess following on from that, do you see, obviously,
is Dave going to do
long experience from working in industry, making decisions as you've said. How's MMIC going to help? And you've given some excellent examples in the sort of cinematic presentation. But looking ahead, how do you see the opportunities for activities like Gavin's maybe described to benefit from the kind of infrastructure ecosystem you have to help on that translation journey?
Yeah, I mean, it's a great question, Alistair. The key thing here is it's got to be industry relevant. At the end of the day, the industry have to pull the technology. If we're trying to push technology to the industry, it's a harder discussion. But if the industry want the technology because they see the value to apply it to their assets, then that's a really important thing. So in the MMIC model, the grand challenges that we've launched and the pipeline that are to come are all industry desired. The voice of the industry is at the heart of what we're trying to do. I think once the industry decide that they want it, then the next question is, how do you bring it to the market in the most efficient, cost-effective way? There's no need for each industry partner to do the same investments, make the same mistakes, take the same risks. If we're prepared to share the risk and come together and trust in a partnership, then you can do the technology pre-competitive stage in a much more efficient, cohesive, and joined-up manner. And really, that's the thesis that I'm presenting on MMIC. And time will judge whether that's actually going to be the case. But I go back to this fundamental question I said in my talk. How can we be 10 years down the line for CDC, but yet we've maybe got a dozen products in the market with that technology? It makes tablets 30% cheaper than the old process today. What's not to like about it? So for me, Alistair, that's the secret there. If we end up pushing a technology solution and trying to fit it in, it's a harder discussion. We must work with the industry and hear their voice.
Great, thanks, Dave. Gavin, you want to?
Yeah, so I would agree with Dave. So that's one way of getting it in. I mean, obviously, I sit very much at the clinical trial end, so phase 1, phase 2. So it's a very small adoption. And I think that's realistically what Ian was talking about as well. And I think, you potentially can get it into the university, into the industry horizontally across the levels, but also vertically as, as molecules come into the market. So we've probably got to have a horizontal approach and a vertical approach.
Yeah.
Ian?
Yeah, I want to build on that. I mean, one of the problems is that it's a 30% benefit on 5% of the cost in a big pharma model. what's really interesting at the moment is that we're getting almost 60 new drugs a year, and a third of them are from emerging companies. So the whole model is actually changing. Not only more small volume, but now smaller companies, and a lot of those backed by private equity or looking for IPO or whatever. So I think this whole cost-time equation becomes much more significant for that for that segment, and I think the place to indoctrinate, building on Gavin's point, is at the beginning in phase 1, phase 2, and get that established. I'm not rubbishing the big pharma, but I think we need different strategies for different segments, and then pull it all together. And I'm feeling quite optimistic that if there ever was a moment in time to be doing it, this is the moment in time, and we're finally there. And I think often you can have great ideas, and if you do it in the wrong era, in the wrong environment, it's like pushing water uphill. But I think we've got a tailwind, and the water's flowing the right way.
Ian, if I can maybe just follow up on that, I think, you know, the innovations and developments you presented for your company, could you have done it using traditional technologies? Is this the only way to achieve the business objectives that you've set yourselves? I think, you know, just in terms of that model, the agility, we've been talking a lot about the slow pace of adoption, and you seem to be fully embracing it. Is that a question of scale, or do you see the absolute technology benefits at the heart of this?
Well, I would say there are three things, actually. Scale is one, technology is another. But the thing we've got on this site as well is we've got people developing drug product across the corridor from drug substance next to bioanalysis and seeing what's actually happening in the patient. And I don't think that exists in many places. You know, I talked to AstraZeneca, and I've talked to many Pengalos and various other people, and I ended up having a conversation with people in California, Sweden, the UK, in order to talk about this. So, you know, I think that's quite important. And in some ways, the COVID crisis might make that easier, in that we have got used to using Teams and Zoom. And maybe we can get away from being separated by miles or hundreds of miles or oceans, and be more intimate on the technology transfer. We would never, by conventional means, I mean, the conventional means are you have the laboratory, you have a kilo shop, you have a pilot plant often away on a chemical facility somewhere else, and then manufacturing a big chemical plant. And what we're trying to do is focus on small volume drugs, maybe high potent, and go all the way from a development environment. That changes the whole cost structure, actually, because you don't have the capital costs, the infrastructure costs, the operating costs of the vast infrastructure to absorb in high volume, high tonnage products. And you can be very much more agile.
No, that's great, Ian. I think, you know, builds and reinforces some of the points that have been raised before around the need to change the approach, the structures of organisations, you know, and the expectations for how, you know, we work around and implement these technologies. One question from the audience, just around implementing end-to-end through the supply chain, requires implementation of reliable release testing. What examples or what approaches are you aware of or considering in terms of helping to improve that critical step? Don't know, Ian, do you want to comment first?
Yeah, I'll just comment on a kind of real example in Arcanova, where just before I took over the site, we would be supplying drug product to a clinic in Leeds for a customer on a different continent. And if they changed the dosage during clinical trials or the formulation. It was about a 3-month process to go through all the rigor you talk about. And that was re-engineered to a state where we can actually be clear on the new formulation during the weekend, and we'll have drug up patient by the following Friday. And I don't think we've lost anything on the rigor. We've thought about how to do it differently, which is a bit like what's happened with vaccines.
And I think the power in those examples and sharing it helps to really emphasise the benefits that can be brought from those approaches. Dave, and I'm mindful of time and that we are between everyone and a short coffee break. So maybe just finish with a question to Dave. Dave, you mentioned around the importance of the triple helix. I think that's been brought through strongly in a number of discussions. Just reflecting on that, accelerated development through the clinic into commercial supply. The MHRA BIA conference last year, somebody talked about the quadruple helix and bringing the voice of the patient in. I just wonder if that's something you see as important, what the potential opportunities and benefits might be of understanding and listening to the needs and wants of patients, as well as, the manufacturing organisations and others in the ecosystem.
It's a great question. I mean, we've talked to this for a while, haven't we, about demand-led supply chains or patient-led supply chains. And if you look, because we're on the topic of clinical supply, but I mean, today it's taking us 6 to 12 months from the time the patient is really wanting the medicine. It's taking us 6 to 12 months to get the clinical product to the right site for that study, typically. And Grand Challenge 2 is a great example of trying to change that paradigm where we're actually treating the packet of medicine that we make as an individual batch. But to do that, you've got to bring on board a real-time release strategy. And the QP dashboard, the release strategy we've got in Grand Challenge 2, we brought the experts from AstraZeneca, GSK. We've got 4 or 5 technology companies working with us. And what's really interesting here is that no individual part of that group of 6 could do this on their own. And it's all about the data architecture and how you bring that data and present it in the right way, connecting the data and a clear strategy for it that allows you then to do real-time release at the end. We actually believe with the PACE technology in Grand Challenge 2, from patient demand, we can get that medicine to the patient within 2 weeks. And that is a transformation in that clinical supply chain. No more over-manufacturing waste and real access and speed into the clinical study. That is transformative, but it is a clear partnership model and a digital and data strategy to do that. And of course, we're speaking with the MHRA. We've shared the concept with the UK regulator, and we're in discussions about how we take that forward. And at the moment, it's hugely exciting.
That's fantastic. Thanks a lot, Dave. I think it's a really inspirational point to end this session on, and just see, the sweep right across the TRLs in terms of innovation, development of new technologies, through to how those are today being implemented in the manufacture and supply of medicines. So hopefully that sets us up well for the next session on regulatory and policy after the coffee break. So I'll draw this session to a close. Thank you to all of the speakers for your time and efforts around this session and contributions. And for everyone, I can ask you to go and grab a coffee. We've got until 15:30, so 3:30. Clive will start the next session. And please do keep the questions coming through and your responses to the polls. Thanks, everyone.
Thank you.
Thank you.
Thank you.